From Microbe TV, this is TWiP, This Week in Parasitism, episode 230, recorded on February 23rd, 2024. I'm Vincent Racaniello, and joining me today from Fort Lee, New Jersey, Dixon Despommier.
Hello, Vincent, and everybody else. I can still see out because it's getting lighter and lighter as the spring approaches, and it's still a gray, overcast, kind of grotty day.
From St. Augustine, Florida, Daniel Griffin.
Hello, everyone.
And from Glasgow, Scotland, Christina Naula.
Good evening, all. Spring here too, but I had some hail coming on my head today, so not perfect yet. But
Oh my!
Our guest today is from Cali, Colombia, from, I'm going to say this, the Centro Internacional de Entrenamiento e Investigaciones Médicas.
Got it.
Dr. Maria Adelaida Gomez, welcome to TWiP.
Thank you so much. I'm super happy to be here. Thank you for the invitation, Vincent and all.
And Christina, thank you for connecting us.
you're very
Your
welcome.
friend of you guys have known each other for years, right?
Yeah,
Oh,
we
yeah.
have. Yeah.
It has been like 12 years or more, mostly.
Yeah. I think you've known Richard first, and then I came on the horizon. I don't know, many years, yeah.
Many. Yeah.
Well, if you like our programs, our science programs, we'd like to have your support. You can go to microbe.tv/contribute. So this program is all about you, Adelaida.
Spotlight.
Spotlight on you. It's okay. We'd like to start by finding all about you.
Sure.
Like where you're from and your training and so forth. So go back as far as you can remember.
Oh my God. Okay. So I was born in Colombia many years ago.
Which city?
I was born in Medellin.
Medellin. Okay.
Yes. And then my parents moved to Bogota, which is the capital city. And that's where I did my undergrad in microbiology in Universidad de los Andes. And then from there, I remember, so since this is part of my story, I'll tell you with a little bit of detail. When I studied microbiology, when I finished, I knew that I wanted to be a virologist. That is really what, at that time, the major Ebola outbreaks in Zaire, I think it was in Zaire, were happening. And I remember seeing in the Discovery Channel these people all dressed in blue in the BSL-4s, and I really wanted to be that. And I tried to look a little bit for training in that, and that never happened. And I got the opportunity to participate as we call it here in Colombia, Young Investigator. This is a program that happens in Colombia where you do not get a degree, but then you have the opportunity to do 1 full year of devoted research without necessarily getting a degree. And when I got that opportunity, I was told that I was going to be part of the parasitology lab. And I almost died because I hated parasitology. I really did. Like it was, I remember in my class, I had an amazing teacher, but what I remember were these life cycles that are tremendously boring. And I said, no way. So I initially started very frustrated. And then I got into the lab of an amazing person that is no longer with us, called John Walker. And he did molecular parasitology, and he did a lot of proteomics. And that's where I got the bite. And from there, so I did the 1 year as a young investigator, and then I went to do my PhD training in Montreal at McGill. And I was there for about 6 years. And I came back to Colombia. I did my postdoc in this institution where I currently am. And since 2011, I have been coordinating the molecular biology and biochemistry unit here at CIDEIM.
Adelaida, I was going to ask, when were you up in Montreal?
I was in Montreal from 2003 to 2006.
Oh, okay. All right.
Yeah, that was a nice time. And now that you talk about weather, just please, I come from the tropics, okay? This is like this weather all the time. And I
Mhm.
arrived to Montreal. I had never seen snow, never. And in August, so just think about this very naive individual arriving in Montreal. August, I thought that from one day to another, winter will come. And it was me, a 23-year-old, looking all around Montreal to try and find winter boots because I thought that winter was coming immediately and I was going to be unprepared. So I know. Yeah.
So it's very hot there right now in Cali, right?
Yeah, we are 33 centigrades at this moment.
Wow.
Oh,
Yeah.
Yeah,
We're
here
at
it's
the
in New
drought.
York. In New York, it's 9 centigrade.
Okay. Yeah, no. Weather here is nice these days.
But Montreal gets very hot in the summer and then very cold in the winter.
Yeah, the weather that is good in Montreal is actually just basically 2 months, the beginning of spring and the
Mm,
beginning of autumn. Otherwise,
yeah.
you get hot and humid and
Yep.
cold
Yep.
and
Yeah.
yeah, too cold.
Well, so you said you went to study microbiology. Why did you do that? How did you know you wanted to study microbiology?
That's a nice question, also with a funny answer. So when um, so I had this boyfriend, special friend, let's call it,
Mm-hmm.
in school, and he didn't know what to study. And I remember that from my high school years, I liked very much microbes, like infectious diseases and this type of things, just because of TV, I guess, and because
Mm.
of these movies that talk about epidemics and those things. And I had in my mind very clearly that if you want to study microbes, you have to study medicine and then pathology. Okay?
Mm.
That's what I had very clearly, evidently. And so we were going through this book to try and see what we needed to do, what career was my friend going to study. And we were going one by one through the descriptions. And then I saw the description microbiology. And the description was exactly what I wanted to study. I'm like, oh, this is it. I don't need to study medicine and then pathology. Oh my God. It's just so naive. It's incredible. Naive in Colombia, naive in Montreal, all around. But yeah, that's how I decided to study microbiology.
We have all had similar stories. I'm not kidding because I became a parasitologist by accident. It was
Uh-huh.
the only job available. If I hadn't got that job, I would have gone to Vietnam.
Wow.
So I was not prepared to go to Vietnam and didn't want to. And I probably would have ended up in Montreal. Bet you. Uh, but fortunately, I got a job in a parasitology laboratory doing diagnostic work.
Wow.
So, we all have these accidental stories to tell.
Yeah.
It's a charming, and you love every minute of it too, right?
Exactly. Exactly. Yeah, yeah. And sometimes when I get to tell these stories to people that are just starting, I really go back in my mind and I remember this feeling of really wanted to be in the BSL-4 and be in all those activities. And now I think back, you see me in the field, literally. This is where we go. We go to the endemic areas, but nothing that has to do with a BSL-4.
So can you tell us a little about CDEIM?
CIDEIM.
What is it and what goes on there?
Yeah, so we are a biomedical research center.
Mhm.
CIDEIM has been conducting research since the 1960s, I think, about more than 50 years. And we are an institution that is devoted to infectious disease research, all the way
Mhm.
from basic research to applied. So we do basic translation and applied. And more recently, we have started to do implementation research and community-based research. And our main focus has been neglected tropical diseases, although we have had periods of time in the institution where we've worked very strongly as well in other infectious diseases like nosocomial bacterial infections. But in principle, we are interested in tropical infectious diseases. And
So,
this is a research center, but it's also a training center. So we do both research and capacity building in research, especially.
mhm. But you don't have students per se, right?
No. So what we do is we partner with many different institutions,
Mhm.
academic institutions, and then it's actually a nice thing because we get students from very different places and they end up gathering here from different universities within the country and outside as well. So it's nice because it's kind of a mixed environment of different thinking processes, let's say, like thinking, yeah, uh, schools.
Can you comment on the general state of science in Colombia? It seems, I mean, I'm not familiar with it, but Cali is not the only city that has research, right?
No. So research in Colombia has grown, I have to say. And I actually experienced it since I returned from Montreal. That was like 15 years ago. Um, it has grown in the sense that a lot of people that have been training outside are now coming back to the country and developing their research careers here. And uh, it's a nice feeling because now you can, as an investigator here, you can feel within a community that actually is able to support the research in a high quality level.
Mm.
Having said that, the research environment in terms of financial situation is very complex. So funding, as everywhere in the world, is very scarce. I'm not sure if it's kind of, yeah, no, I shouldn't say that. It's scarce. And for example, in our case, we depend mostly on grant funding institutions that are international, NIH, Wellcome Trust.
Yeah.
So it's a bit difficult.
So the Colombia itself doesn't have sufficient funds to support research, basically.
No, not that much. Not that much. It's very little of the percent budget from the country that it's devoted to research. There's not a lot of interest, I have to say, unfortunately.
And what other cities have research activities?
Where, you mean?
Yeah, in Colombia. Yeah.
Oh, so you mean in which cities?
Yeah.
So in the major cities, definitely in Bogota, which is the capital, in Medellin, in Cali, Cartagena, Santa Marta, Barranquilla. In many major cities of the countries, we have
Okay.
universities that have either research centers or the professors within the universities do some research. But again, it's a little bit different than the US, for example. And that's, I think, why I have a privilege to be in a research center. I can devote 100% of my time to research,
Right.
but people that are in universities here have almost 60 to 70% of their time devoted to teaching and only 30 to research. So you cannot really do a lot of research like that.
Yeah. So besides your laboratory at CDM, what else is? How many other labs are there, and what do they do?
Within our research center.
Yeah.
So we are, it's not the typical arrangement of labs that are specifically focused on one pathogen or one theme. Essentially, our whole institution works jointly for different for different infectious diseases and different projects. So in CIDEIM, we have research on Leishmaniasis, which is what I do. We have research on STIs, especially syphilis. We have had large projects on the immunobiology of congenital syphilis, for example. We have had a history of doing quite a bit of malaria research. It's not that active at this moment, but that's something that we have done quite a bit. Dengue, TB. I'm thinking, oh, well, COVID. I mean, everyone has touched COVID, but essentially vector-borne infections and STIs.
Right.
How
So
about,
what would you,
I'm sorry.
go ahead, Dick.
How
Let
about
me just ask one more.
Go ahead.
What is the most prevalent infectious disease in Colombia?
Oh my God. I mean, I have no idea.
Be malaria?
No, I mean, parasitic.
Any
we're
infectious
any
disease.
infectious disease, I am tempted to say maybe respiratory tract viral infections. Not sure
Okay.
exactly which, but we have a lot of prevalence of those and a lot of prevalence of gastrointestinal infections as well, but associated with malnutrition primarily.
Mm-hmm.
And then in the parasite side,
Right.
we have a lot of malaria. We have a lot of Leishmaniasis, Chagas, you name it. All of it.
Okay.
I was going to ask about cysticercosis. Do you see some of that also?
I no.
Because
But
Venezuela has a lot of cysticercosis.
we have, but I'm not too sure. So we don't do that type of research here. And in terms of the epidemiology, to be honest, I don't know.
Okay.
So I may be saying something that is very wrong.
So we're going to go over one of your papers in a bit, but maybe you could give us an overview of the interests of your laboratory.
Sure. So we work with human cutaneous leishmaniasis, and one of our main interests. So this is a program that has been ongoing for 40 years. It started, the Leishmaniasis research program started with Nancy Sarabia, who is our scientific and actually the director of the institution. And at the beginning, what it tried to do was to characterize the epidemiology of Leishmaniasis in Colombia because it was unknown. And that happened, let's say, in the 80s, more or less. A lot of descriptive work and a little bit of mechanistic work in the context of natural history of infection was done at the institution. And all that generated a very strong background knowledge that allowed me to do what I currently do. And so I have been very interested in understanding the basis of treatment failure. And for me, so my passion is host pathogen interactions. Okay, this is what I really love. And the way that I have tackled the therapeutic responsiveness is by understanding host pathogen interactions, essentially. And the work that we have been doing for the past 15 years has been mostly focused in understanding what is the contribution of the host to treatment failure in an infectious disease, which in this case is Leishmaniasis. And it has been a very special road because your mind, or my mind at least, has changed a lot of paradigms that were here. Treatment failure is because you have drug resistance in your bug. No, not necessarily. Host is important, but not that important as the parasite. And really what we're starting to see is that the host response is really, really determinant in defining a good therapeutic outcome or a bad therapeutic outcome. So that's essentially, in a nutshell, what we've been doing, and then tipping on the sides in terms of mechanisms associated with chronicity of Leishmania infection, of different disease manifestations, a lot of training as well in parasitology and in research, like building capacity in the country and in different communities on how to conduct biomedical research with human subjects. And that's the other thing. The work that we do in the lab is 100% associated with patients. So we don't do any animal work. We do just patients with human studies, and then a little bit of functional validations with cell lines and stuff.
So do you concentrate on just one species of Leishmania, or do you have a broad-based?
So we do broad, but broad in the context of what's important for the region. So in Latin America, most of the species that cause cutaneous leishmaniasis are of the Viania subgenus.
Mm-hmm.
And these are particularly interesting because those are the ones that are able to cause mucosal disease.
Spondia.
Yeah. And so that's also another quite interesting subject of research that we are pursuing, and it's the host pathogen interactions that allow the parasite to persist in these different tissues with or without causing pathology. So just let me tell you a little bit very quickly. So people that have actually cutaneous leishmaniasis, if you go and you test in their nasal mucosa or in the conjunctiva or in the tonsils, you can find by molecular biology, by RNA, RT-PCR or PCR even, you can find the parasite there. And the parasite is there years and years and years. And one of our interests of our actually current projects is trying to understand what are the mechanisms that allow the parasite to persist in those tissues.
Adelaida, can I ask about that? So is
Sure.
that people that will then later go on to mucocutaneous, or that's just everyone and only a subset will actually develop disease?
The second is true. So,
Wow.
well, I cannot say everyone, but in our studies, for example, we have seen that about 40% of people that have cutaneous leishmaniasis that are treated and then heal, if you go and you check back in their mucosal tissues, you find the parasite.
Wow.
And then, unfortunately, these studies are very difficult to do prospectively. It's difficult to follow them up for 20 years or so. But in older studies, about it changes depending on the endemic area that you are, but sometimes about 5%, maybe less, can have recurrence. So it's really a minor proportion of the people that actually have clinical manifestations in a persistent infection.
And these are all cutaneous species,
These are all
not
cutaneous.
Brasiliensis.
So Brasiliensis is cutaneous. So we work with Brasiliensis.
Well, we call that mucocutaneous, where we always say mucocutaneous.
Okay. So in the region you have Brasiliensis causing cutaneous, Brasiliensis causing mucosal, and Brasiliensis causing mucocutaneous.
Oh my.
You have also Panamensis, same thing, and then Guyanensis, same thing. So we particularly concentrate on those three species, and also Leishmania mexicana, amazonensis, those others that do not cause that many cases here, but that also circulate in the region.
Now, let me ask this. Please go, Christina. Then
Sorry.
I've got another question.
Okay, me too. I was just going to go back to that persistence that you mentioned. Do we have an understanding of how long that persistence lasts? Is it months? Is it years? Decades? Or do we even know?
So listen, that's a very nice question. I think that's lifelong. So this is not because we have done a prospective study, but years ago we did an epidemiological survey to try and demonstrate anthroponotic transmission in cutaneous leishmaniasis in the Americas, which has not been proven. Okay, but that was the purpose of that study. And in that study, we surveyed different communities and were able to detect parasite persistence in people that reported that, let's say, a person that was 60 years old, and they reported that have had disease when they were 5-10 years old. So we know the oldest person that we know is a span of 52 years from the moment that had the infection to the moment that we were able to redetect the parasite. Now, what that means, what it does to your immune system, is it protective? Is it actually a risk factor for other infections? That's really what we want to discover. I mean, I'm excited because that's a new project that is ongoing. So,
and do we know where the parasites persist? You know, particularly in which tissues?
So we find it in nasal mucosa, definitely. We find it in conjunctiva. We find it in tonsils. It has been found in adipose tissues in cadavers. And I bet you that we can find it in the gut, but you don't search it in the gut at all, because why would you want to do a biopsy? I think in almost all mucosal tissues, but that's not proven. Okay, it's proven definitely nasal, conjunctiva, tonsils, adipose tissue. But we're interested particularly in the persistence in the nasal mucosa, because that's where we more frequently find it.
So I was going to circle back to, so let me get this. So the big three with mucocutaneous potential, right? So Brasiliensis, Panamensis, Guyanensis. they're doing this, they're there, and we're just waiting. Only certain people are going to get pathology in those mucocutaneous sites. But colombiensis, does it go there but doesn't cause disease, or it doesn't go there? I'm just sort of trying to.
Oh, you got me on that one. To be honest,
Well, if
I
I've got
don't
you, then
know.
maybe nobody knows, Adelaida.
It's just it was such a random. I mean, in the years that I've been here, we have not typed a single Colombiensis, and
Okay.
we receive around 400 cases every year. And with the colleagues in the north part of the country, I don't think that they have typed a single Colombiensis. I think it was associated with a cutaneous lesion, but to be honest, it doesn't circulate that much, or whether it's just a subpopulation of one of these vianias, I don't know.
Wow.
I'm sorry to say it, but I don't pay that much attention to that one.
You ignore the other ones.
Yeah.
The one named after your country, we'll just,
That's right. That's right.
I'm not even going to ask about Mexicana. We'll just leave it.
Did you ever
That
have
one is actually pretty bad.
Did you ever have the pleasure of meeting Philip Marston?
No, I haven't.
I actually talked with him for some time.
Ah, nice.
Yeah, and it was amazing because he would talk and talk and talk. As long as you wanted to listen, he would talk. And he had stories about the Trans-Amazonian Highway being built and the
Wow.
emergence of disease all the way along until they got to Brasilia.
Listen, that's an amazing story. I mean, it touches my heart right away because I just finished reading La Vorágine. That's a book that essentially talks about the story of the rubber plantations in the south in the Amazon region, and it brought me back exactly to that. And in the past years, there has been a lot of enthusiasm in Colombia to bring people to the jungle areas for ecotourism, and I've had the pleasure of going to these places, and just the feeling of being in the middle of the jungle, being really vulnerable to all of these vector-borne diseases and many other threats that you cannot even imagine. Oh, that I would say that 2 or 3 bottles of wine are not sufficient for a night talking about that.
So, Adelaida, before we jump into your paper, maybe you could give us an overview of the infection cycle of
Beautiful.
Leishmania.
What actually brings us all together, the infection cycle. So, Leishmaniasis is transmitted by the bite of a sandfly. That's a small fly. And essentially, what happens is that when you get bitten by the sandfly delivers the stage that is motile, and that's the one that we call the promastigote. That stage rapidly gets phagocytized by monocytes, macrophages, neutrophils, especially. And then within the phagocytic cells, it develops into a non-motile form, which is called the amastigote. And that amastigote is the one that is able to replicate within the cells and is able to continue the cycle within the mammalian host. Then if you are in an environment where there are sandflies and the sandfly bites you back, then the sandfly can pick up some of your infected cells, and it continues the life cycle like that. In this area of the world, we find a lot of other mammals that are infected with Leishmania, especially dogs, opossums, something that we call ant bear. I don't know what's the name of that in English, but it's a bear
That's the
with
same
a
as
very
an
long...
anteater.
An anteater.
Exactly, yeah, that thing.
Sloth?
Sloths as well, indeed.
Yeah.
3.
They're so pretty, yeah. And in the Americas, what I was saying at the beginning of our conversation, in the Americas, it has been essentially the dogma is that the transmission is zoonotic. So we go first through a mammal before going to the human host. But there are so many areas where you put traps for mammals to check for infection, and they're all negative, but you have a lot of incidence of infection in humans. That's why we were actually interested in trying to prove that there wasn't trophonotic transmission. But unfortunately, when we went to the endemic site, there was no transmission at all. There were only 3 cases in 5 years. I was so sad.
Oh my! What about primates?
Primates, I'm not sure. To be honest, I don't think that. I mean, they are able to get infected and develop a lesion, but usually those are self-healing. Actually, here when we do animal captures, primates are not the target. I'm not sure if it's because of low prevalence or just difficulty in capturing these animals. But in the ones that have been found the most are the opossums and dogs, definitely.
Okay. Why don't we dive into your paper? So this is a
Sure.
Journal of Immunology. Consolidation of a molecular signature of healing in cutaneous leishmaniasis is achieved during the first 10 days of treatment. And so you're the senior author here. Maybe you could tell us first, who are these people and what did they do?
Sure. These people are amazing. So I'm very proud of our research team. This is just part of the research team. Lina Giraldo, she's the first author. She's a research assistant in our lab. She did her master's degree here and stayed with us as a research assistant and has been here with us for about 5 years. Then David Rebellon, he's a physician and he's currently doing his PhD at Imperial College. And in Colombia, different than the States, for example, physicians that are interested in research are very, very few. And so this is one of my happy moments because these people really got engaged with research and decided to support their career with a PhD as well. And so he's now there. And he supported everything that had to do with patient recruitment and a lot of data analysis. Adriana Navas, she was a research assistant in our team working with me for the first 10 years. And then she decided that she wanted to do her PhD. And she went on to the Netherlands and she's currently finishing up her PhD there, but she worked in all of these initial stages of the project. Then Trey and Najib, well, Trey and Najib are my collaborators from University of Maryland. Trey is a research associate in Najib's lab, and Najib has been my research partner for like 10 years now. And he is side by side with me supporting all the transcriptional analysis that we do in our translational research projects. And then it's me.
Yes, we know you. So what is the problem here that you're looking at?
Yeah, so this is the issue of treatment failure again. So for cutaneous leishmaniasis, the main treatment that has been used for decades are pentavalent antimonials. So pentavalent antimonials, and this is the way that I try to make it close to people usually. Antimony is essentially the cousin of arsenic, and everyone knows what arsenic is. And antimony has exactly the same properties in terms of toxicity as arsenic. And this is the drug that we use to treat cutaneous leishmaniasis. It's a systemic drug. And with this drug, if you have full compliance to the regimen, we have about 30% of people that do not respond well to treatment. And so there is a big problem of treatment failure, a big problem of toxicity, and what are we going to do with these people that fail treatment? So in our research, what we want to understand, as I was saying at the beginning, was is the host-related determinants of therapeutic outcome. And why is that? Because even though drug resistance in these parasites has been demonstrated and has an association with the outcome of treatment, it is not the only determinant. So essentially, you can have people that are infected with drug-susceptible parasites that then do not respond well to treatment. And in the contrary, you have also people that are infected with drug-resistant bugs that respond pretty well. And that is really the conundrum that we want to understand. Then the other little piece of problem that we have here is that patients with cutaneous leishmaniasis in the Americas are patients that inhabit rural areas. And just to give you an example, we work with a lot of patients from the Pacific coast of Colombia. These patients have to travel by boat about 5 or 6 hours to be able to go to the health post to have diagnosis. And diagnosis is necessary for treatment. So that means that if you have a patient that is diagnosed and then it's treated, the patient has to stay outside of their place for about 20 days to be able to be fully compliant to treatment, or travel 5 hours back and forth every single day for 20 consecutive days. And the outcome of treatment is not defined as soon as you finish the therapy. So this is 20 days of treatment, but at 20 days of treatment, the lesion is not healed. So you have to wait 3 months after you finish up the treatment to make sure that the person healed or not. So this means that the follow-up of these patients can take 3 months, 6 months, even a year to make sure that they have a good clinical outcome. So with this particular research project, what we wanted to capture was the changes in the cutaneous lesions over the course of treatment in patients that have a good outcome and in patients that have a bad outcome, and understand at which point during treatment the major changes occur so that we can identify potential biomarkers that allow us, one, to be able to modulate the inflammatory response to promote healing, and second, to be able to build or identify potential prognostic biomarkers to be able to say with more certainty the patient is going to have a good response or not, so that we don't have to do the long follow-up and then reinitiate treatment so many months after. In fact, in these areas, and essentially this happens throughout South America, the people that comply with follow-up is less than 1% of the people that are treated. So you lose everyone, essentially. You don't know what happened. Most of them most likely would heal, but some of them may not. So it's important to have these things that you can capture the patient, not capture the patient, but capture the phenotype as early as possible.
Adelaida, could I ask what is sort of the protocol for failure? So you go ahead with your frontline therapy, person fails, and then what is sort of the second line?
This is a bit absurd, but this is exactly it. It has changed a little bit in the recent years, but the traditional protocol is you have pentavalent antimonials, you fail. Okay, then you have a second round of pentavalent antimonials, you fail. And then you can have a third round of pentavalent antimonials, and then you just die, essentially.
Yeah, we call that insanity when you keep doing the same thing over and over and expecting a different response. But so there's no way of liposomal amphotericin, sort of a miltefosine combination approach. You're just hitting over and over with the same.
Yeah. So no, actually, what I'm saying is something that happens in the clinic, but I think more and more that this problem is visibilized, then people are a little bit more conscientious of not prescribing a second time antimonials. So usually no, but in some places, such as a reference center like ours, what we do is a patient fails, then most likely the second line drug would be miltefosine. Amphotericin B is not good for cutaneous leishmaniasis because it doesn't have a good penetrance in the skin. So it's very good for mucosal, it's good for visceral, but not good for cutaneous. And then the thing that is, it has been kind of a gift in a way, is that our local guidelines have changed recently to allow local treatments. So we can now use thermotherapy and intralesional antimonials, for example, for treatment, but that's only used for certain cases, people that have just a single lesion, lesions that are below a certain diameter, 3 or 4 centimeters, depending on the guideline that you follow. And about 50 to 60% of the cases are not compliant with the requisites that are needed for local therapy. So you still need to give systemic drugs.
Mm-hmm.
What happened to the use of ketoconazole?
No, it's not really used. So there have been some trials with several different azoles. It's sometimes, I'm not sure if it depends on the region where you are that it works well or not, but the experience here is not that great. We have actually a really nice paper that was, I wasn't one of the authors, but it came from CIDEIM, doing some preclinical analysis of azoles in combination with miltefosine, and it was really nice. In vitro, it looked really nice but in ex vivo models. But we haven't done clinical trials to check for those combinations. It was a combination, if I recall correctly, of posaconazole and miltefosine.
So tell us how you address this problem of treatment failure in this paper.
So what we were doing here, in the past, I think a lot of these studies that have tried to characterize the immune response in the context of treatment have looked at systemic responses under the assumption that these are going to trace what happens in the tissue. And a couple of years ago, we did that comparative analysis between what happens in the skin and what happens in PBMCs. And although the responses are similar in the sense of having, let's say, pro-inflammatory responses associated with treatment failure, what you observe in the lesion is much more complex, and it involves many other pathways that are not found in PBMCs. So that's why we decided we just go to the lesions and characterize the lesions to see exactly what's occurring at the site where you have the clinical manifestation. And so the way that we designed this study was that we conducted a prospective analysis of patients with cutaneous leishmaniasis, where we took lesion biopsy samples before initiation of treatment, at the middle of treatment, and at the end of treatment. And we decided to do a transcriptional approach, a transcriptomic profiling essentially, to try and capture as much as we could from these lesions. And that was complicated because the lesions of leishmaniasis are ulcerated, necrotic. They're like nasty lesions. So one of the things is, how are you going to get a good transcriptome from a lesion that is all necrotic? That was one of the main things to optimize. And so essentially what we do is we take lesion biopsies that are in the lesion border where you don't have necrotic tissue, but you still have a lot of inflammation, and a little bit of the healthy tissue, just to make sure that it doesn't become a mush once you take it. And so Lina, who is a very talented research assistant, she did all the optimization of that methodology to be able to have good transcriptional profiles of those lesions. And then essentially what we did was we followed up the patients. We had some patients that had good therapeutic outcomes and others that didn't have good therapeutic outcomes. And we did a descriptive comparative analysis of these lesions and how they change over the course of treatment. And to me, what was one of the things that was really striking is that if you ask a clinician, for example, and they are looking at the lesions of these patients, but midway of treatment, these lesions are still ulcerated. They are not healed by any means. And so you still have a very active ulcerated lesion, but you have a transcriptional profile that is very associated with wound healing or inflammation. So very early on, before any commitment of, before any clinical evidence of healing is observed, you have a molecular pattern that is associated with healing.
And these patients were local patients, or were they distributed throughout the country?
So these patients were people that came to our headquarters in CIDEIM in Cali, and also patients from the South Pacific area of the country. So that area that I was commenting to you about, that had to come hours and hours in the boat to be able to arrive. So those are our two places where we conduct our research.
So did you collect samples from, so you're describing 16 patients in that paper, but did you, initially, obviously you weren't going to know what they were, whether they were going to have treatment failure or treatment success. So did you collect samples from many more patients?
Yes.
And then decided on the 16 that you wanted?
Yeah, so because treatment failure is about 30% of the cases, this was a cohort of about 60 patients. And then we just matched to the treatment failures the cases of treatment success. And, it's matching in a way, because sometimes, for example, a patient says, I don't want to donate another biopsy sample. So you have a lot of sample sets that are incomplete. Sometimes you just lose the patient to a follow-up. So you cannot have, you don't know the outcome of treatment. So there are a lot of additional situations that are out of your control that end up, from a very large cohort, reducing the cohort to a small cohort like the one that we analyzed. But this initiated with a group of 60 patients.
So you find that the patients who didn't heal, you see downregulation of genes involved in keratinization, right? So downregulation from the state before infection, correct? Because you have those samples beforehand, right?
Yeah. So what we see is not necessarily there is a slight downregulation, but it's not. So let me try to explain this before. What happens is that inflammation continues constantly. It is really never resolved. And in addition of inflammation being continuous, there is no particular pattern of wound healing or keratinization at all. So we see downregulation of some keratin genes. Some of them do go up a little bit, but never as much as what we see in the patients that respond well to treatment. But really what I think it's really the major contribution to treatment failure is the sustained inflammation that we see in these patients. Not only the lack of keratinization, but also the continuous inflammation, which I think is kind of the loop that avoids the keratinization.
So somehow the inflammation is suppressing keratinization, do you think?
This is what I think, This is what I think is happening. Not sure what's the mechanism, but I think inflammation is what's dampening keratinization.
You don't think that the parasite is involved in any way in, say, dampening keratinization?
Well, that's actually a good question. So that's a discussion that I have with friends and colleagues. They say yes. I tend to say no. Why is that? Because when you trace the parasite is eliminated, or not eliminated, but it's the parasite load is almost undetectable as early as 3 days after you give the treatment. So if we go and check for parasite loads in these patients, the parasite loads midway through treatment are almost undetectable. So I don't think that there's a lot happening with the parasite, really.
Okay.
I think, though, that there may be some antigen persistence that promotes this constant inflammation. But I'm not sure how. I cannot explain that.
Adelaida, was
Leishmania
there
has a
any
double-stranded.
hint from families that there were certain families that were more likely to have issues non-responding? So was there sort of a population background?
Yeah, so we have not done any of that. We tried to do a GWAS study, but because it is so random treatment, it's so difficult to find treatment failure, we have not been able to do any family studies. I can tell you that in the GWAS, but this was, of course, a tiny little GWAS study that is not even published. This was with 150 patients, something like that. We haven't been able to find any gene at the significance level. At a very high, we have like -6, -5, but not at the GWAS significance level. So it has been difficult to pinpoint. There have been some clues that there may be some genetic variants associated with chronicity or mucosal disease. And maybe in the context of inflammation, this may be associated with outcome, but we have not been able to find it.
What about the double-stranded RNA virus? Infects some Leishmania, but not others. Have you looked for that?
Yeah, so that's also something that we have looked and looked and looked and looked. So I can tell you, this is also a paper that we published recently. We took from our biobank about 150 strains of Leishmania panamensis, which is the strain that we mostly analyze, and we have not been able to detect the parasite in any of those strains, in any. We detected in Brasiliensis, we detected in Guyanensis, but not in Panamensis.
Yeah,
we looked in the strains in this study.
Yeah, I'm just thinking it would be so ideal instead of waiting till day 10 and then you say, oh, look at this different. If it's really a host factor, just knowing ahead of time. Like, you're not going to respond. We're going to use this different approach.
Yeah. So I hope this is the paper that I'm hoping to submit this week. So literally, I have my colleague, harping on him. Can we just do the submission letter? But we did a very nice transcriptomic profiling looking for predictive biomarkers. And this was transcriptomic profiling that we did in monocytes, eosinophils, and neutrophils. And we actually find that type 1 interferon responses are predictive of treatment failure. So what I think is happening is that you have a lot of inflammation mediated by type 1 interferons that is positive, looping and looping and looping and looping. And we hope that we can then go and use this and, do a validation trial, not a validation trial, but do actually the biomarker trial. Let's see if that works.
Yeah, because it is. So
that's
when
preempting the paper that is not even submitted.
I was going to say, so when I work with people up in Panama, it is quite something to leave your community, all the expense, the travel, go to somewhere, be there for 3 weeks, and then have it fail.
Exactly. Exactly. It's in terms of public health, it's a big problem. And you just imagine this with a child. A child has to be with a parent. So this is not just one person moving. It's the mom or the dad and the child having to move away. And actually, this is some of the things that catch my heart, because I remember I am a microbiologist. I'm not a physician, but I remember the first time that I saw a patient. This patient was a large man, like very healthy indeed, and he had a cutaneous lesion in the knee, and his job was in agriculture. And the problem that he had was that the boots that they use in agriculture here in the tropical regions are boots that are rubber boots that go up to the top of your knee. And so the boot will always hit in his lesion. And this was a perfectly healthy man. And it was so painful. He had to leave the work that he was doing. Therefore, he couldn't get the daily wage to feed the family for a month for a disease that is not supposed to be that problematic. So and actually, thank you for that comment, because this is a very clear example of the social impact of these diseases that are not deadly, but are still very neglected. And those are the things that we don't think about. It's not because it doesn't kill you, it's because it really has an impact on the quality of life and in people that have difficult access to healthcare, to just economical sustainability. This is what happened with neglected tropical diseases.
Yeah, and then you imagine that
So
individual
Daniel
goes,
is,
gets treatment, and then it doesn't heal, right? So you're just like, oh my.
Exactly.
Daniel, is there any role for steroids in this uncontrolled inflammation?
It's almost the opposite. What we'll do a lot of times, I think Adelaida brought this up, and I think we've talked on other episodes, is people will try to destroy the local tissue where the parasites are, right? So the hot machete, maybe the local plants that actually, like a poison ivy type response, you rub it, and then you rub it a few weeks later, get a blistering. So we're often trying to destroy the tissue around with the parasites. Yeah, it's an interesting issue. Like, what would happen if you start putting steroids on one of these areas in the tropics? I don't think it's something we've done, but yeah, I'm not sure, because what we're seeing here is this failure to decrease the neutrophil activation, the T cell activation. If there was some way to modulate that, maybe that can allow things to heal.
Yeah, but modulated in the sense of dampening. We want to dampen for sure the very strong pro-inflammatory response, the one that is not it's the uncontrolled pro-inflammation or inflammation that continues. So I think, I'm not sure if this is a tricky question, because I think the important thing there is when to use your anti-inflammatory medication or whatever immunomodulator. And as far as you control your parasite load, that you do not exacerbate it by using an anti-inflammatory drug, then you're fine. So if we're able to decrease the parasite load to a point where it's not going to reactivate, and then we can control inflammation, then that's, I think, what could be done.
Yeah, maybe like the timing we've learned from COVID, right? You start your anti-parasitic, you get 10 days in, then maybe you might add a period of some sort of, and what would be the right anti-inflammatory, I'm not sure. Yeah.
Exactly.
All right. Anything else, folks, before we say goodbye?
This is fantastic work.
I have a million questions, but we don't have a million hours to go through them. But I would love to sit down with you sometime, and maybe you could come to an ASTMNH meeting.
I would love to, for sure. Absolutely.
The next meeting is in New Orleans, so it's pretty close to where you are, actually.
I had to move it, though, because it conflicted with my Taylor Swift tickets. But
they changed the date of the ASTMH.
Well, I thought I was going to do both, right? But apparently, no, I have got to go to New Orleans twice.
So you're going to have to go twice, yeah, because I thought that was a really weird, I saw it on X, that the ASTMH had been moved because there wouldn't be enough hotel space available.
That's right.
Because of all the Swifties coming to New York.
That's right, all the Swifties. They were faster than we were, that's for sure.
You have tickets?
Who, me?
Oh, to the Swift? To the Swift?
I have relatives.
Yes.
That's a personal question. But anyway, I will be at ASTMNH.
Right, okay. Okay.
And so I guess I will too.
All right. That's TWiP 230. You can find the show notes at microbe.tv/TWiP. Send your questions or comments to twip@microbe.tv. And if you like our work, please consider supporting us. Microbe.tv/contribute. Our guest today from CIDEIM in Cali, Colombia, Dr. Maria Adelaida Gomez. Thank you so much for joining us and telling us about your work.
Thank you so much. I'm super happy to be here, and welcome to this beautiful country that has nasty diseases.
Thank you, Adelaida, for coming along.
Thank you, Christina, for the invitation.
Daniel Griffin's Columbia University Irving Medical Center, parasiteswithoutborders.com. Thank you, Daniel.
Oh, my pleasure. Everyone, be safe.
Dixon DePalmier, trichinella.org, thelivingriver.org. Thank you, Dixon.
Oh, you're muted.
Dixon, you're muted.
But we won't tell him.
Well, I had a great time, and I really enjoyed hearing all about your work, and I look forward to looking at your next paper.
Christina Nowell is at the University of Glasgow. Thank you, Christina.
Thank you. I had a lot of fun and I've learned lots of new things. And it was just lovely to catch up with Adelaida as well.
I'm Vincent Racaniello. You can find me at microbe.tv. I'd like to thank the American Society for Microbiology for their support of TWiP and Ronald Jenkins for the music. You've been listening to This Week in Parasitism. Thanks for joining us. We'll be back in 2 weeks.
Another TWiP is parasitic.
All right.